And now we come to our final speaker this evening, who is Angelika Schnieke. She is the Chair of Livestock Biotechnology at the Technical University of Munich and Dean of the School of Life Sciences there. Angelika is also a fantastic and incredibly honoured person to have with us because she was working for PPL Therapeutics, which is the company set up by the Roslin Institute to work with their cloning technology. And she was there at the time that Dolly the sheep was born and made first, then born. So thank you very much. It's really great to have you here, Angelika. Thank you.
Well, thank you very much for the kind introduction. And of course, it's always a pleasure to come back to Edinburgh. And it's a great honor to talk at a meeting where such eminent scientists as Ian Wilmot and Professor Yamanaka are talking. So I was asked to sort of give my personal recollection, how I got involved in the Dolly story. And well, like most of the good things in life, it starts with a good cup of tea, or in this case, it was probably one of the famous chicken pies, which were at the Roslin canteen. So it was one lunchtime, we were sitting there, Alex Kind and Jim McGuire, and we were just talking about experiments we were doing, and he was saying, well, we're doing something really exciting. I'm just doing something with Ian and also with Keith. I have cultured some cells, and not for very long, but I could keep them in culture, and from these cultured cells, we actually made some sheep. At the time, they only had the pregnancies, and later on, these two sheep are Megan and Morak. So these were the sheep which Ian mentioned, which actually came before Dolly. And that he had cells in culture and could still make a sheep, that was what excited me. As you heard already, we were working at the time, Alex and I were working at PPL Therapeutics. And what we were doing there is we were producing drugs, pharmaceutical protein, in the milk of transgenic animals. But to do that, we had to go through a long procedure. So the only method we had to make transgenic animals were DNA microinjection. For regulatory reasons, we first had to make mice. And only if the mice were working and we could milk the mice, which is possible, but not very easy. And of course, you don't get much milk out of a mouse. Only if you proved that that was all correct, then we were allowed to make the sheep. But to make transgenic sheep is even less efficient than making transgenic mice. That meant we had to sort of have a birth of about 100 sheep to maybe have 5 transgenic and maybe 1 or 2 which really expressed our genes at a very high level. So when Jim said that they did now, together with Ian, the cloning from those cultured cells, then we thought it would be actually really great because we already had a project going on where we worked with mammary cells. So if we could actually put our genes into mammary cells and then see, is the gene there? Is the gene expressed? We get a little bit sort of as something similar to milk out of the mammary cells, which you can keep in culture. And if you could then use the cells to actually make an animal out of it, then all we need, because we have done all the tests in cell culture, all we would need is 1 sheep and not 100 sheep. So that would have been, of course, very nice. Actually, the experiment that we get milk out of the cell culture hasn't quite worked. But I went to our boss at the time, at PPL, and was really excited and just said, look, they're doing these exciting experiments at the Roslin Institute, and that would really fit with the project we are doing here. So what do you think? Can we go do cloning maybe with the adult cells or cloning at all? Could we, of course, if it would be adult cells, if you have adult cells, yeah, mammary cells that had to come from an adult, now this would overturn a dogma, a biological dogma, which had before, as Ian explained, in frog it was called, you can do it with the embryo, you can do it with fetal, but you can't do it with adult cells anymore. So that was exciting because that would maybe also have an effect that we didn't have to use quite so many experimental animals, and it might also allow us later on to do precise genetic manipulations to go into the genome and change things directly into the genome. But unfortunately, the boss, yes, he was a bit excited, but first of all, he said no. He knew the experiments from Gurdon, and he said, this is much too risky. I will give you financial support that you can do some cloning experiments, but you have to use the embryonic cells. And so we got the financial support from PPL Therapeutics to do 3 sets of cells, which had to be embryonic cells from those sheep which we were working with for the milk production. So what you can see there is, for example, an embryo, and where you see the tiny arrow, there's sort of a little dark dot. And this dark dot is the actual cells which later will make the embryo. So you can see there's not very many cells. To get those cells into culture, to make sure that those cells survive for a long time is not very easy. So we tried several times, mainly with Jim's group, and we had in the end a few cultures, but unluckily in my experiment I had a little bit of a hiccup, which was in the end a good thing to do. Some of the colonies got contaminated. Not quite as bad as what you can see here, but they weren't quite good enough to use anymore. And then finally our boss Alan Gender said, okay, instead of just now using 2 of the embryonic cell lines, you can go ahead and do the adult cells too. So it was really a lucky thing that we had a contamination, that in the end, in that experiment, we were finally allowed then to do the lucky experiment. And if you really be honest, out of all these cells which had been, or the embryos which have then been generated by the nuclear transfer and have been replaced, only 1 of them survived. And it could have gone exactly the other way. You know, in the first experiment, we did the embryonic cells, Ian's group did fetal cells, and we did the adult cells, and from all of those we got animal born. It was a great time, and you can imagine, as soon as the publication came out, it was an absolute uproar in the scientific world, and also it was a media frenzy, you could say. That we had lots of people who were excited, we had lots of people who were critical, and lots of people who also said, what have they done? Now, what will be the next step? We had also lots of people who were skeptical, because it was a dogma which was overturned. So they were saying, no, this can't be right, you have done some sort of trick. And so we actually had to do forensic science. So we had to get people in from outside the labs who took completely independent probes from the cells which we had, from the animals which were in Ian's group, from Dolly, and really prove that the only way this animal could have been produced was through the nuclear transfer. So, and of course, we also had other people like my children. They could just only say, what is all this fuss about? Don't we have enough sheep in Scotland? One more, what does it really matter? But I think what the real excitement also was about Dolly is that you had people talking about science all over the world. We were shortly afterwards, we were on a holiday in India, and it was really just on a small boat somewhere in a backwater, and you could hear people were talking about Dolly. Dolly really sort of brought an excitement back into science. Of course, because of all the inclination, could you do it in humans? Are you actually even allowed to do it in animals? There was lots of work now to do for ethics, so they had a lot of jobs. And to really be honest, it also helped us to get our jobs and more prominent positions. And for me, what I'm very proud of is also I became a famous Scottish woman. You can hear from my accent that I'm probably not Scottish, but it was just in the Scottish women's or lesbian calendar, I can't quite remember. And normally the famous women are allowed to have their name behind their birthday. For me, it was the birthday of a sheep. It would make me a little bit younger, but unfortunately that's not the case. And very often when I get introduced for talks, I'm introduced as, here's Angelika Schnieke, she is the mother of Dolly. So being the mother of the sheep is maybe not quite so exciting as being the mother of dragons, and I know which one my husband might prefer. But what Dolly then also meant for science, and that's really what we also should talk about here, is the experiment we could do then afterwards. So in 1985, we had the first transgenic large animal which had been produced. So that was the first genetic modified. Then 10 years later, Dolly came across. So she wasn't genetically modified, it was really more a reproductive technology at her point. But we could show then a year later that we can actually make transgenic animals. And I will explain to you also that this makes the whole procedure a lot more efficient. And then 2 years later, we could also do gene targeting, so precise modifications. And then over the years, all these other animals, which Ian already showed you, could also be cloned. So here you have the procedure very briefly, and I don't want to go into the details because Ian was talking about it. What I just want to point out is that How does it make it more efficient? Cloning is not the most efficient way. We always have still losses. But we can now take just any sort of material from an animal, some tissues. If you have adult animals, you have a lot of tissue. We, for example, work a lot with fat. We work with pigs, they have a lot of fat. Or we sometimes also get bones. We can just get this material from the slaughterhouse, get these cells into culture. We do then all our genetic manipulation in the culture. We test that we have the right clones, and then use those sort of pre-selected clones to make our animals. Where do we get the ovaries from? Originally, they had to come from the sheep, of course. But now we just go to the slaughterhouse and collect the ovaries from the slaughterhouse, isolate our oocytes, which we need from that, and then mature them in vitro, and then the nuclear transfer is carried out. So this whole top side of the experiments is all done in vitro, so we don't need any sort of live animals for this. And only when we then go into our pseudopregnant mothers, so when we do the embryo transfer, that's the only time where we actually then go into live animals. So that means by using the cell culture system, we can actually refine our experiments. We can already do pre-selections. We have a replacement because we now work mainly with slaughterhouse material, and overall we reduce the number of animals. Remember I told you at the beginning we had to have almost 100 animals born. Now principally one animal would do. So one healthy live animal, and we can do our experiments. And I mentioned that we could also hope not only to make transgenic animals, also to do gene targeting, so precise genetic manipulation. We could do it in the mouse for a long time. Just like Professor Yamanaka has mentioned, we had embryonic stem cells. And embryonic stem cells are very good to do homologous recombination in it, but also very good afterwards to take those cells and make whole animals out of it again. Or now you can also do it with the induced pluripotent stem cells. Both of it works. But in the livestock animals, we're still not very good at isolating ES cells, and the same for the iPS cells. It's still difficult, and I would still say we don't really have functional cells. But Dolly gave us the opportunity to do the experiments anyway. Whoops, sorry. So here's an experiment now. In my own lab, we work a lot on animal models for very serious diseases, for example, for cancer. And this is an experiment in pigs, so we don't have embryonic stem cells. What you can see there at the top is a gene which is called the adenomatosis polyposis coli gene, which I hope you now all will remember. But this is the gene which causes, in most cases, colon cancer. And the bars at the very top show you mutations which are very, very common in humans. And if you have those mutations, you will get lots of polyps and then eventually also colorectal cancer. So we took now exactly those mutations, introduced them over homologous recombination into the porcine genome, in the somatic cells, produced them the animals by nuclear transfer. These experiments were done in collaboration with Eckhard Wolff. And then we got the animal, we get the disease phenotype, and what is very nice to show is that all the polyps are in the large intestine exactly the same phenotype as what you see in humans. But it's different from the mouse, where if you do the same mutation there, you would have it in the small intestine, so in the other part of that. So it works, and it works also with the large animals, and the large animals seem to be a good model. And there is a lot of large animal models which are now being produced, and almost all of them are now being produced with this nuclear transfer technology. So why do we do it? Because we think that a lot of procedures, of course, can still be done and are done in the small animals in tissue culture as much as possible. But it's a big step to come from a small mouse to a human, and a lot of the experiments fail when you come to human patients. And so the larger animals are a good model for preclinical tests, for validation, for safety, and to just bridge this gap between the tiny mouse and the large mammal, which are the humans. And another experiment which is possible, it's, of course, pigs are agricultural animals, and you can get your sausages. I come from Bavaria. We have about 5 million pigs which are slaughtered every year to make our sausages and our ham. But it's maybe more important that you can actually also take organs, hopefully in the future, from the pig to actually save lives. And This was a recent Nature publication, which they said with new technologies, this should become possible. And one of the technologies absolutely was Dolly, because it's such a difficult project. You have to see that if you would take a Picard and put it into human, it would be rejected within minutes. That means you have to bring in not only just one transgene, we have to bring in multiple transgenes, and we also have to inactivate genes which are there in the pig and not there in humans anymore. So it's something which happens through evolution. And with the Dolly technology, we can actually do this. And we now actually have animals which can survive in, not in humans yet, but in monkeys for almost 2 years. We're not there yet, but it might be the future. And of course, it's not always only the human health. We should also think of improving the animal health. And again, with the technology which comes from Dolly, with the cloning, but also with the new technologies which now come in, like genome editing, these molecular scissors, which makes it even easier to do precise manipulation. We can also improve the animal health. There's just one experiment mentioned there at the top, and I also know that at the Roslin Institute, they're also working on particular animal diseases. So here we have new technologies like this genome editing, the molecular scissors, which now you can do directly precise manipulation in the embryo. So principally, you might not need the cloning technology anymore, but for some experiment where you not only want to destroy a gene, where you want to replace a gene, where you do more complicated manipulation, it might still be worth to do it first in the culture, test that you have the right cell clones, and only then you make the animal. So, and with that, with the outlook in the future, I will stop. And I must admit, I mean, Dolly doesn't look quite so healthy anymore, and apparently you can all go to the museum and have a look at her. But I still think, even up to today, she is something which has inspired scientists and is still inspiring scientists. And for my own, I can really say, well, I have the chair of livestock biotechnology, and without Dolly, it would not have been possible. Thank you all for listening.
Thank you. If we have 1 or 2 questions just for Angelika. We'll take a question there in the middle.
Thank you for your talk. So you were talking about preclinical models in animals, right? How do you know which animals will be good to recapitulate the same disease in humans?
Before you do the experiment, principally you don't. So you can have a good guess. We know that the physiology, of course, of a pig is much closer to that of a human. There's also the genetic differences between humans and pigs is slightly smaller than between a mouse. So when we started, for example, this one experiment, we had no idea if we really see the difference. So you have to do experiments to see which it is, and probably no animal model will ever be perfect. The perfect model would be the human, but of course you can't do this. But it has a lot of advantages.
Thank you.
Any more questions quickly for Angelika?